a pleasure to be here, and particularly in a year when epidemiology has been in the news basically every day. Um, yeah, I imagine all of you have become very popular amongst your social circles in the past year. So, as Siddhi mentioned, my PhD is actually in computer science. So I have actually, as it turns out, no medical training whatsoever. Uh, that said, I've done a lot of learning, been very motivated by a lot of patients to do so. So what this really boils down to is if there's anything in this talk that resembles medical advice, please don't take it because I'm not qualified to give it. And the truth is, even to this day, it's obviously becoming less true with time, most of my academic career was still on the computer science side. I spent a lot of my time doing cutting edge applications in cybersecurity. I did a fair amount with high performance supercomputing, and then a lot of just fundamental algorithms and data structure discovery. Some of it related to AI and formal methods within computer science. So that's still where the bulk of my training and academic experience has been. But really, for the past 5 or 6 years, I spent most of my time focused on precision medicine at the biggest scales, for national initiatives, and then at the level of individual patients as well. So I want to make a claim about precision medicine and really where medicine in general is headed. And this is that when we look back at the history of medicine for the 21st century, we are going to find that data was in fact the greatest drug we invented during this time period. And what I mean by that is that not that data is a drug, but that the use of data and as a medical intervention, I think will outweigh any other major innovation we make over the course of this century. I really do believe that. And this is relevant for me in the sense that precision medicine is this new field which essentially uses data as a drug or converts data into a drug or allows us to better treat patients with data. And I do take this very deliberately broad definition of the field. And you can get to almost every other definition of precision medicine from this one, including things like personalized medicine. So, well, what is precision medicine at a high level? If you go all the way back to the start, I think the first definition of it occurred in this National Academies report back in 2011. There's a small essay in the glossary on what they mean by precision medicine and whether it's personalized medicine or whether it's something else or whether it's accurate medicine or precision medicine. But what it really boils down to is this. If you are using the individual characteristics of the patient to do treatment, then to drive tailoring of treatment, then you're doing precision medicine. That's really what it boils down to. Now, of course, those individual characteristics come in the form of data. So data can drive this really personalized tailoring to do precision medicine. So I think the most encompassing definition I can come up with is precision medicine is about optimizing health with data. And occasionally I get some pushback from frontline clinicians saying, well, I've always been trying to optimize my patient's health. How is this any different? Well, I think what's different is two things. One is the amount of data available, both about the patient and outside the patient, and the complexity of the optimization process itself, reaching the point where it really starts to necessitate computational intervention in the process. So I like to visualize this as a very high level process. And on the one hand, as the input, we have the patient or really all of their data. On the other side, we have some sort of optimal treatment for them, which could be a medication, could be lifestyle, could be environment, could be a combination of all of these things. But the piece of data that got folks really excited about precision medicine was in fact the human genome, which became clinically accessible about a decade or so ago, maybe 12 years, I think, is when it really started to become accessible. And I think it was, the human genome that forced, medicine to reckon with the fact that, it's so big that you really do have to rely on computational aid to interpret it. And there's other forms of big data or big enough data that the same is true as well. And when you look at the state of clinical informatics, you realize that, we really haven't even, tapped the full potential of just what's sitting in the electronic medical record. So that's another form of data that can come to bear on this optimization process. And then there's all the data sitting in books or on Google or elsewhere, all the biomedical knowledge that's ever been published. All of that should be part of this optimization process too, when it comes to finding the right solution for the patient. So I do think of it as essentially forming this constrained optimization problem with all the data you can get on the patient and trying to identify the right combination of, let's say in this case, lifestyle and treatment to achieve the optimal health for them. So in a nutshell, that's what we're trying to do in precision medicine is identify the right combination of inputs to give us the best possible output for each patient. Um, and there's a process for this. And roughly these days, there is certainly even before this a precision prevention component to it, which I'm not going to get into too much detail today, except to say that if this is something you're interested in, I highly recommend looking into the Alabama Genomic Health Initiative, which has a major precision prevention component to it. From what I do, I like to focus on the diagnostic and especially on the treatment side of precision medicine. So there's sort of a two-phase process for how this works, how you find that best treatment for the patient. In the first phase, you're trying to get to a root molecular cause. Um, and rare diseases happen to be relatively right for this part of the process because genomic sequencing is very powerful at jumping straight to the root molecular cause in many cases. But in principle, a lot of diseases have molecular causes and layers of causes. And so any of those can become hooks for the precision therapeutics process as well. And in general, we want to go back as far as possible in the sort of chain of molecular causality as we can and intervene as early as possible. But any mechanism of harm is fair game for driving precision therapeutics. And within precision therapeutics, once you have your hands on a specific mechanism that's driving disease, when we pull them apart, we really ask, in which direction is this mechanism going? Is this mechanism overactive, in which case we need to inhibit it in some way? Has it become underactive, in which case we need to activate it in some way? Has it become absent, in which case we need to compensate for that absence or replace it in some way? Or has it changed its behavior in such a way that it's become toxic, in which case we need to eliminate it? And if you don't remember anything else from the rest of this talk, I think this is the key takeaway about precision therapeutics, because for every patient that we deal with for this process, this is really the thinking that goes into it. What's the mechanism, what's gone wrong, and how do we reverse the impact of that harm? So we do have a program now. In fact, we have medical directors as of January at the Institute that oversee a sort of, if you will, a precision care clinic. And really what we're doing there, it's mostly rare disease patients that we see, but we're trying to apply this process to anybody that's been on an intractable diagnostic or therapeutic odyssey to see if we can find a way to use their data to find the right treatment for them. And I do want to just sort of clarify a few things, because sometimes people will conflate, precision medicine and genomic medicine, and they're definitely related. And in fact, if you look at the landscape of precision medicine today, it is dominated by genomic medicine. And that's just because the large piece of data we're dealing with most frequently is a genome of some kind. And so they're reasonably good synonyms these days, but this is shrinking over time. And I think in the future you'll see genomic medicine as a small but central and very important part of precision medicine. But as other forms of data enter the picture, so we have new initiatives now that deal with, not just transcriptomics, but metabolomics and proteomics and all sorts of other big data modalities that give us our hook into potential mechanisms of harm in a specific patient. All of these are shifting, the way the field is operating, and also expanding the scope of the diseases that we can more easily access with the technology that we use. So I of course had to wonder, well, what does this mean for epidemiology? So I'll tell you a little bit about, and some of this you likely already know, what's been going on at this intersection. But it's clear to me that epidemiology is a very data-driven field. And so some of the techniques that we use in precision medicine for our optimization, or even just the sort of generic application of artificial intelligence or machine learning, certainly would have applications in this field as well. So I do recognize that epidemiology is not just infectious disease, but it's certainly a very visible part of it. And I can show you how you can grow from the N of one perspective on infectious disease up naturally into a population perspective on infectious disease. So, for example, we have helped individual patients using metagenomics when it comes to infectious disease. And your metagenome is just you plus all of the organisms inside of you and their genomes as well. And if you have an infectious disease patient, you can take a sample, you can do raw reads on the sample, sequencing reads on the sample, and what you're going to find is DNA fragments for a whole bunch of organisms. Most of it's the patient, 99 point some percent of it's going to be from the patient themselves, but the leftovers are telling you what else is floating around in the patient too. Um, and we have used this in the past actually to generate plots like this for a specific patient to say, this is what's inside them. Now, it turns out in any one point in time, there's a lot of organisms inside a patient. And so the challenge becomes, well, how do you find the one that's probably driving the disease in this particular case? Of course, a lot of these are, perfectly benign. Some are perfectly safe as long as they're in the gut, but they all show up here. That's the problem. So what we've done for individual patients is fuse metagenomic data with top-down predictions based on medical records and phenotypes and things like that to arrive at a set of possible diagnoses. So we can say, well, here are the bugs that we see. Here's what AI says this could be based on observations on the patient. And hopefully somewhere in the middle is what it actually is. And certainly that has been the case before. One of these cases was written up in Stat News about 1.5 years ago for how we did this in real time. And if you want to know more about that, you can certainly read that article. But this also starts to get us more towards, precision epidemiology. And the crazy part about this is that, it's being talked about in sort of a somewhat hypothetical sense, 2 years ago, and in February exactly, in fact, pre-COVID, we said, well, it should be possible, if there's a pandemic or epidemics to start doing this sort of genomic, tracing of infection. And sure enough, COVID came, and that's exactly what's been happening. And it's become extra important once again with the emergence of all these new strains to do genomic surveillance as the disease mutates and evolves. And so the thing is, we can see it almost in real time as these strains arise, and really a chain of who infected who. And so we've been in talks about pooled testing strategies and things like that with companies here in Alabama about how to do genetic contact tracing. So when you start to pick up on a pocket of COVID infections, you can try to figure out who infected who within that pocket and where is it coming from, things like that. And just to say a word on COVID-19, we've certainly done a fair amount for this. We stopped most of our patient-facing operations for the better part of 1 year and focused almost exclusively on COVID-19 for a little bit. We've done lots of drug repurposing. I'm not going to cover all the collaborations we've had in this space, but we did most of our focus in this time period was on repurposing existing drugs to help treat COVID. We helped launch an RCT of anti-androgens for severe COVID patients with the VA. And then more recently, we published this paper just using our own EHR data, in an observational way to identify, drugs that seem to be helping at-risk populations, and found very strong associations between metformin and reduced mortality for diabetics. This has since been backed up by some molecular and in vitro data, and even some in silico data we've been able to generate. So it's all been feeding on its, we have a number of ways of approaching repurposing, which I'll get into. And a lot of it sort of builds on its one modality will build and work on another modality. So, for example, if we see a trend in medical records that appears to be beneficial, we can try to construct a molecular story, and then validate it in vitro to see if there really is something there. And that, of course, puts us in a good position to then recommend the launch of something like an OT. So what I want to focus most of the talk now on is, I think that one of the things that UAB is doing sort of uniquely well, and even sort of uniquely trying to take on, which is precision therapeutics. You'll find plenty of places that are doing precision diagnostics. There's a lot of really good undiagnosed diseases clinics out there today. UAB happens to have an excellent one as well. But I think one of the interesting, to really unlock the promise of precision medicine, you've got to be able to benefit the patient at the end of the day. And that's where I spend the bulk of my time. And it's where a lot of the Institute invests most of its research energy. And I really do think of precision therapeutics as a step-by-step process. And because it's a step-by-step process, and because I'm a computer scientist, I now think of it as an algorithm, the algorithm for precision medicine. And I'll show it to you in high speed and then break it down through a series of examples. So patient data comes in, we try to find that root molecular cause, usually through sequencing if it's a rare genetic disease. And then we're back to the core of the flow chart I showed you earlier, where we're looking to check, is it a gain of function? Is it a partial loss of function? Is it a total loss of function? Is there some introduced toxicity? And hopefully, once we work out all the strategies, each of those directions gives you a different strategy for drug repurposing. Once you've worked your way through what the appropriate set of strategies, hopefully you've arrived at candidates for end of one clinical trials, or maybe even in some cases, larger scale clinical trials. And I'll talk about cases where we've done N of one, and cases where several patients have gone into actual clinical trials as well. So there are several ways to go about this process of identifying repurposable drugs for a given patient or for a disease. I mean, when it came time to work on COVID, we essentially took all of the technology we've been developing for individual patients and we repurposed it for one disease where the molecular mechanisms were in fact well understood. So all this technology, while we apply it mostly on a day-to-day basis to individual rare disease patients and increasingly to oncology patients as well, it could be repurposed itself for anything, any disease. And so we also have projects in Alzheimer's, we have projects in diabetes. It's not just rare diseases anymore. So, but one way to do it is to actually start testing drugs. And by that, I mean, we start with a patient and we pull from them some sort of disease avatar or model of that patient's disease. And there's several ways to do this. You could take, patient tissue itself. In some cases, that's a reasonable thing to do. You might create a cell model. Maybe you transfect in a mutation into a cell line, and now you've got a human cell line that's not from the original patient. But increasingly, we're actually building up animal models. And in fact, we have I have a large new award here at UAB with Brad Yoder for this center we've created that does nothing but precision animal modeling. And what that means is we have a center where physicians or patients or scientists can reach out to have a precision disease model made of a specific disease. And so we have several model systems that are in scope at the center right now. You can do C. elegans, you can do zebrafish, you can do mice, you can do rats, you can do xenopus, and we'll be expanding this over time as well. There's more expertise on campus than just those systems, but these are the ones we wanted to start with. And once you've got the model, you need to phenotype the model. Once you've got the phenotype, then you can start testing drugs. So in a repurposing drug screen, we'll just test all approved drugs against the model and check to see if any of them rescues. For all approved drugs, there's several libraries you can use to do this. One library includes effectively all approved drugs that are both FDA approved and approved somewhere in the world. It's about 1,500 drugs. And there's another library, which is a bit larger, but it starts to throw in natural products and also tool compounds. It's about 2,600 compounds. So, these are, pharma wouldn't consider this a high throughput screen, because high throughput for pharma could mean millions of compounds as they do drug discovery. But I still think of this as a high. It's not 4 or 5 drugs. It's a few thousand drugs that you're going to test to see if it works. And so, in some cases, you can do this with a postdoc or a graduate student or a lab tech, and in some cases, you do want some automation to help move through the drug screen. And if you do find something, then the next step is to figure out, okay, well, for this drug, will it get to the right tissue? So is delivery a solved problem here? And then, more often the blocker is, but does it work at an appropriate concentration? So you have to look at something like the IC50, both in the the asset that you have, and also what's tolerable biologically. So it is frequently the case that you will find an approved drug that has the effect you're looking for in a dish, but then you realize that to achieve the same concentration in a human body, you would kill the patient. That happens a fair amount. So medicinal chemistry will come into play at that point for trying to get the potency or the needed concentration to somewhere where that's biologically reasonable at that point. The other way to do drug repurposing and for individual patients is through artificial intelligence, and this is one of the major focus areas of the institute. In fact, up until we got this new NIH Center for Precision Animal Modeling, this was the primary way that we did it on a day-to-day basis, and it still is the primary way that we do drug repurposing. But increasingly, we do have this model organism capability as well. The tool we built to do this, which is freely available to anyone, it's open source, and anybody who needs access to it or wants access to it can email me. It's called Metagenomics. It's been funded by the NIH through NCATS and a program called Translator. And what it does is it's really fusing together good old-fashioned logical reasoning with all the biomedical knowledge that we can get our hands on. And there's a reasoning engine in the middle called Mini-Canron, which is why it has the name Metagenomics. So it's artificial intelligence in the sense of the 1980s view of artificial intelligence, which is to say it was more logical than statistical. And this actually has some big advantages. And one thing that differentiates this from the logical AI of, let's say, the 1980s is that the amount of structured biomedical knowledge that we have access to now dwarfs everything we had back then. So just as with statistical AI, which has really enjoyed a renaissance in the form of things like machine learning, the data is once again what really changed a lot of what we can do with it, just the sheer amount of data available. I think the raw count of the number of facts sitting inside this biomedical knowledge base that we have, it's into the low hundreds of billions of facts, structured facts that it has memorized at this point. So what we have in this AI tool is really a combination of old school logic with high speed automated reasoning to achieve superhuman-like deduction with clinical insight. That's what we're trying to achieve with this tool. And you can ask it very low level questions like, hey, what's an inhibitor for X, this specific gene, protein, enzyme, you name it. How do I slow it down? Or you can ask it very high level questions like, hey, what may be a treatment for X, where X could be something like overactive bladder. And it can scour a number of data sets and come up with suggestions. So for overactive bladder, in particular, it can come up with 145 different strategies that might be in scope for overactive bladder. And more importantly, it can explain every one of these strategies with references to the medical literature or other medical databases. The disorder that actually earned us the funding for this tool was Fanconi's anemia. So we were part of an NIH hackathon. There was a challenge to say, can you find a novel treatment for this? There were Fanconi's anemia experts in the room, and when we turned the tool loose and started to really pick it apart, here's what it found. So we realized that with Fanconi's anemia, you can't repair double-stranded breaks in DNA. These double-stranded breaks are primarily caused by reactive aldehydes. These reactive aldehydes are generated by things like alcohol dehydrogenase, and you can inhibit alcohol dehydrogenase with compounds like xylitol. And so it created this chain of reasoning to say, well, maybe xylitol can ultimately, 4 steps later, slow down the introduction of these double-stranded breaks in DNA. And it found about 10 other compounds that can do this. The judges were quite impressed. And so that's how we got the initial tranche of NIH funding for this specific tool. So how do we do this? Well, all we had to do is read 30 million papers for the first iteration of this. And of course, we didn't read it. We used natural language processing, another form of AI, to help condense all these abstracts in PubMed into structured facts. And so if you look inside the brain for Metagenomics, here's what it looks like on the inside. It has a whole bunch of nodes. And each of these nodes is a noun. It's a concept. It's a gene. It's a drug. It's a symptom. It's a disease. It's a protein. It's a molecular variant. It's something like that. And there's relationships between these represented by edges. And these are predicates that connect the nouns together, that form full relationships that create effectively sentences. So that you know that some gene has a specific relationship with a drug, or some drug has a specific relationship with some gene. Um, as an example of that, you have Prevacid inhibits NGase. So that's an example of a fact that lives inside Metagenomics' brain. And this is how that fact gets represented. Again, the nouns are on the nodes, the relationships are on the edges, and then you can encode all sorts of biomedical knowledge this way. You can also do reasoning between nodes this way. So for example, if you know that blue, whatever it is, increases red, whatever that is, and red decreases green, you can infer that by increasing a decreaser, um, you should ultimately cause a decrease. So you can infer that blue should decrease green as a matter of logical inference. And so you can start to connect dots in the literature that don't exist within a single publication or a single data source, but that ought to be there. And so Metagenomics' brain is really just a soup of these facts and relationships all mixed together like this. And then you can query across it. In terms of taking this process to patients, we've been developing a research consultation service. It's recently crossed the line into essentially a beta tested clinical service, if you will. So we do have medical directors now. We do, in some cases, directly see patients, though mostly what we're doing is we're consulting with physicians. Yeah, certainly plenty of patients reach out directly, but what we prefer to do is to engage with their physicians and come up with a game plan based on our understanding of the condition. To scale this up, we also have a secret ingredient. It turns out that secret ingredient is just people, but not just any people. Sitting underneath the clinical team is this engine, made out of brilliant students, mostly undergraduate students, as a matter of fact. And a lot of the initial heavy lifting is done by these students. And just to give you an example of who they are and the kinds of things they've done, I'll walk you through some of the things they've figured out as part of this program. And on a day-to-day basis, what they're really doing is they're acting as go-betweens between the physician, the patient, the AI tools, and in many cases, scientists as well. And ultimately, when a recommendation goes back, it doesn't go back, from them to the physician. They may draft the initial report. It's going to get vetted by our medical directors, and then it might go to the treating physician. But still, a lot of the hard work was done by them. And so I think they're an integral part of this process. And yeah, one of the things they do particularly well is use Metagenomics on behalf of others, whether it's another clinician or a scientist in some way. They're an important part of this process. The other thing we do is we have case reviews every Monday where we as a team review every patient that's reached out, and we take a look at what we might be able to do for them. To make this an efficient process, we have developed our own EHR, if you will. It's completely de-identified, so it just exposes the relevant scientific details for each specific case. And what this allows us to do is, since everything is totally de-identified, if you want to join in on any given Monday, it's actually happening right now. I'm stepping out of that. You're welcome to. And we'd love to have you drop by and observe and engage in this process. And I will say that, everybody who's come in with some piece of expertise has inevitably ended up contributing to it in some way, because there's, such a huge diversity of cases. We see that almost any expertise ends up being relevant at some point for one of them. We also have a lot of great faculty and staff that help with this process as well. I just want to give a shout out to some of them. In fact, I need to update this slide because we have quite a few more that have come on board lately. And now what I'll do is just walk you through some of the patients that we've helped using this methodology. So for example, we had a patient that had a variant in a gene called TMLHE. May Crumbly was the undergraduate analyst working on this at the time. She reasoned that this was a loss of function in this particular gene. This gene happens to be involved in carnitine biosynthesis, and so should lead to a deficiency in carnitine. And the remedy then was supplemental carnitine. In this case, the patient happened to be suffering from seizures. And after a 30-day course of supplemental carnitine recommended by the patient's physician, saw a substantial reduction in the number of seizures. So that's an example of how an undergrad armed with some of these tools could end up writing a report that ultimately influences clinical care. Another case handled by Jillian Tinglin, it illustrates some of the complexities of what can happen in a space like this, and some of the workarounds you have to use to end up helping a patient. In this case, it's a genetic disorder driven by mutations in a gene called ROBTB2, results in a large number of ataxic episodes every day, and but which can be treated or appears to be treatable with celecoxib. And here's how you get to that point. With these variants, it looks like they all cluster. If you look at the patient population, they cluster in a specific domain that controls or really enables degradation of the protein. And so what these variants are doing is they're breaking the ability of the protein to be degraded, which in turn results in an overabundance of ROBTB2, or in essence, a gain of function in ROBTB2. If you drop ROBTB2 into any pharmacological database, you drop it even into Metagenomics for the simple stuff, there is no direct known inhibitor of ROBTB2. But ROBTB2 is regulated by E2F1, and that has targets that can downregulate it. So celecoxib, for example, can downregulate E2F1, and since E2F1 expression drives ROBTB2 expression, you can indirectly drive down ROBTB2 expression using celecoxib. So when this report went back to physicians in Brazil, they reported a reduction in ataxic episodes in fact, almost a total cessation, I think, in ataxic episodes for that specific patient. And one of our older cases is from about 3 years ago is MAPK8IP3. This is, since we've been working on it so long, we've kind of become a center of excellence for this disorder as a whole. It's something we do a lot of work on now, have become a hub of activity for the patient community. We helped in diagnosing one of the first patients who came through the Undiagnosed Diseases Network. We realized that this mutation was likely leading to a haploinsufficiency in MAPK8IP3. And Lindsay Jenkins was the analyst working on this at the time. She then plugged this into Metagenomics and found that retinoic acid, or more generally vitamin A, looked like a pretty potent upregulator of MAPK8IP3. And so a research report went back to the treating physician saying, I think you should use, or doesn't say I think you should do this, it just documented the literature around the nature of the variant and papers citing vitamin A as a way to upregulate MAPK8IP3. That physician ended up coming up, prescribing a high but non-toxic dose, of course, of vitamin A for this particular patient. And and right away, I mean, nothing happened. So in this particular case, for this particular disorder, and MAPK8IP3, they tend to be non-ambulatory. In fact, a lot of them can't even stand. It's very difficult for them. This particular patient was 5 years old when they first reached out and went on this recommended treatment strategy. And 6 months later, after months of really nothing happening, we got a video from the mom, which I'm sharing with her permission, showing that this child was actually now starting to take his very first steps. So this is pretty astonishing for us. We definitely didn't expect, we didn't know what to see. We didn't know what to expect really, but this is what we saw. And so this is a child who was effectively stuck on the floor months earlier, and now he clearly needs some assistance in standing, but this is significant progress. And so not only is he standing up, albeit with some aid, he's taking a few steps here and there. And what's amazing is a year later, we got another video, which I'm just going to share with mom's permission as well. And it's same child, but now he's definitely making significant progress in his walking. So again, 18 months prior to this, he couldn't really even stand up. And now he's taking, very concerted steps as he navigates this room. And you'll see in just a second, not only is he capable of walking on his own, but if he gets down to the ground, he can actually get himself back up and keep going. So he's making significant progress. And there's corresponding, progress neurologically as well. So he's making strides in terms of communication and in school. And so, it looks like this is working. We don't know, I mean, it seems to be divergent and substantially divergent from the natural history for the disorder.
That was amazing.
And what we're doing to follow up on that is we're actually, trying to recreate this same result using animal models like fish and worms, and also do expanded drug screens to see if we can find something better than vitamin A for these patients as well. And we're sort of working towards a case study. The next case I'll share with you is one you may have seen in the media. This is the first case ever diagnosed of hyperactivity in this particular potassium channel, KCNMA1. And as the first patient in the world with this disorder, we say first in the world because she was the first gain of function patient ever found for this. Not that there's a lot of other patients, but there's a very different phenotype for the disease when you have a gain of function in this potassium channel versus a loss of function. So she's a gain of function, and she was suffering on the order of 150 cataplexic episodes per day with this disease. So it was severe. There was a column in the New York Times by Lisa Sanders that covered her case. And then there's a whole episode of a Netflix series called Diagnosis dedicated to this specific patient as well. We became engaged right around the time of the New York Times article, but also because she was a UDN patient and we're part of the Undiagnosed Diseases Network Coordinating Center. So we engaged early on. As soon as we saw it was a potassium channel, we said, we think that these ion channels tend to be more druggable. Let's see what we can do. So we jumped in, and a year ago, got some results back from Metagenomics, and it very, matter of factly said, well, why don't you just give this child crystal meth? They said it looks like it's like, oh, I predict methamphetamine will be a very potent inhibitor of this particular potassium channel, and sure enough, it is. It actually is. Of course, we looked at what's and just said, well, we can't give meth to a child. We can't do it. So we looked at some of the other stuff that came out of Metagenomics. We got all excited about spider venoms and plant toxins, which also are very potent and selective inhibitors of this particular channel, and thought, let's do that instead. For some reason, rather than finding a way to work with methamphetamine, we decided it was better to work with dangerous toxins and venoms and think about somehow diluting these and injecting them into this poor patient's spine. Now, it fortunately never came to that. We also did a chemical screen, a full repurposing screen, looking for other inhibitors of the channel. That concluded recently. But in the meantime, 3 more patients showed up with this particular disorder. And as a matter of fact, one of them had already been successfully treated. In fact, that patient is now on his way to a pre-med program for college. And I called up the desk and said, well, how exactly did you do this? What was it? And he goes, oh, it was pretty straightforward. It was amphetamines. So amphetamines, and they just by dumb luck stumbled across this. They had no idea, even at the time that they gave this, because this is prior to the diagnosis, that this was going to work, but it did, and it worked spectacularly well. So this was a lesson for us to say that sometimes Metagenomics is too specific, that it should have identified a class of drugs here that rather than one specific drug as being in scope. So we have a structural similarity search and a structural similarity option in the tool now. So you can say, well, I can't give this structure, but find me something similar that I can give. And so if you chop off that methyl group, turns out works just fine. In fact, what we do in practice is an extended release amphetamine at the start of the day, and that seems to keep these cataplexic episodes away. And in fact, I got in touch with the patient group again just a few days ago, and now this treatment is working in 5 patients. So there's now 5 patients around the world successfully treated with this particular strategy. And the next step here is to start bringing them all to NIH. So we're pretty certain that we'll be able to do either perhaps a simple withdrawal trial and show that this drug really is working in these patients. And we understand why too. So Metagenomics, if you ask it why it's working, you'll find that it's an indirect attack on the channel. Amphetamines end up downregulating a microRNA that in turn was responsible for upregulating the alpha subunit on that particular potassium channel. So it indirectly lowers the expression of that channel. We've also hit a new milestone. We've actually taken a Metagenomics prediction all the way to an FDA-approved trial for a group of patients. In this case, it was this mom reaching out saying, hey, I'm looking for treatments for my child's disorder. It's ADNP-driven autism. Turns out it's a haploinsufficiency in ADNP that drives it. At the time, Metagenomics didn't exist, but a year later it did. And we were able to write back to the chief science officer of their foundation to say, we've got an idea and you might want to vet this. So it's low dose ketamine, as it turns out, and it strongly upregulates the wild type ADNP allele. And we said we think this might actually work. And so they spent a year digging into the basic science behind this. They tested this in animal models. They did a lot of cell modeling and convinced themselves, and in fact, clinical trialists at Mount Sinai and the FDA ultimately, that this was a good thing to do. So they set up a clinical trial. It got approved. The pandemic interrupted things a little bit, but eventually got it enrolling. And so I think it was back in October, maybe, they started enrolling their first patients and putting them on treatment in this trial. I'll close out these cases with a non-genetic case to show you that, when you build a tool like Metagenomics, even though you may intend to do one thing with it, it ends up being useful for more than you might have imagined. In this case, with intractable cyclic vomiting, we had a 19-year-old patient with pretty severe version of the disease where she just literally couldn't stop vomiting and was facing multiple episodes per day, was inpatient hospitalized at this point, and was only 78 pounds, despite the fact that she was 5'4". Just could not keep food down at all. And of course, the parents, when they reached out to us, said, look, we've tried everything. And what they really meant is they've tried all the treatments that they knew and that their physicians knew. But, medicine is an expansive and old field. And so it's very likely you've tried everything for some condition, especially one as common as nausea or cyclic vomiting. So amongst the 30 million some odd papers that are out there, 64,000 of them are on treating nausea. That are on nausea in general. 4,000 of them are on treating nausea. And among them, there's 374 distinct treatments proposed for nausea. And so no patient has tried all of them. In fact, if you rank them by the number of publications and support, there's 169 papers for Zofran. So this is perhaps not surprising. This is the number one hit. But if you keep going down this histogram of treatments against the number of citations for them, you descend into what we call a region of the unknown known, where there's only a handful of publications and support, but these are not widely known techniques because they're not even, say, the top 100 techniques used to treat nausea. They're way down the tail. But if there's more than 4 or 5 or 3 papers for some of these treatment techniques, it means it's been done a few times and might actually work. And so we started moving down the list of treatments, and she had, in fact, tried a lot. That is true. But she hadn't tried everything. There was something hiding out in the middle of the long tail, which was nasally inhaled isopropyl alcohol. So essentially just sniffing isopropyl alcohol during bouts of nausea. And recommended this to her team. And sure enough, they recommended it to her. And then she went 8 months in a row without vomiting. And it didn't stop there, actually. It's to this day not returned. It's just that at 8 months, things had changed substantially for her. She was up to 125 pounds. And in fact, her high school sweetheart, who had been visiting her every day in the hospital, proposed to her when she got out, and they got married. So they moved to San Diego a few months later, and that's where she lives now, and lives a life that she just never could have imagined. Now, the interesting thing here is that we never got molecular resolution on this disease. We don't know exactly what's causing it, but we do have a list of every conceivable treatment for nausea. And so when people feel like they've tried everything or they've run out of options, it's very likely that we can generate more options that are substantiated by something somewhere in the scientific literature or in some scientific data set that's out there. And so this is something we've done in a few other cases where people just say, I just don't know what else to try. And we can work with their physician to see if there's other options available for them. So I'll close with my own personal story and why it is that a computer scientist does all this stuff anyway. And for me, it's really focused on my son Bertrand. So this is him when he was about 1.5 years old, at the beginning of a diagnostic odyssey. So we started off on a place that so many other families in rare disease do, called Undiagnosed Island. And when you're here, trapped here, it's really, it's an island of one, population of one patient or one family, and you don't know why your child is suffering. And for the first 4 years of Bertrand's life, we really didn't know what was causing his seizures, his developmental delay, his movement disorder, or his really curious inability to cry tears at all. So he could cry. I mean, he could go through all the emotional aspects of crying, but he would never actually make liquid tears. And this got so bad, in fact, that he had to have multiple eye surgeries to correct the chronic infections or weird like drain pus from his corneas and things like that. It was just awful, trying to manage these horrifically dry eyes on a day-to-day basis. And it got so bad in the middle of the diagnostic odyssey that, they told us at one point that he probably only had 2 or 3 years left, or 2 or 3 years in total to live. So when he was 18 months old, physicians were telling us, based on the lab values they were seeing, that he probably wasn't going to make it much longer than another year and a half or 2 years, if that. But we got lucky. I made it all. We made it all the way to a diagnosis, and it took about 4 years to get there. But he was one of the first human beings to ever have his exome sequenced. And what it revealed was that he was the first known patient of a brand new genetic disease and called NGLY1 deficiency. This launched me in a number of directions, which ultimately brought me to UAB. There's of course a question of when you see a novel set of mutations in a gene that's never been associated with the disease before, you have to ask, how do you really know this is what it is? And there's kind of a standard process we do a lot in genomic diagnostics where we take all the phenotypic data points about a patient and we try to map them onto this underlying purported genotype. And we have to ask, does this genotype explain what we think is what we're seeing in the phenotype? And in this case, and in every case, in fact, you have to understand what the gene is doing. So NGLY1 encodes an enzyme that is responsible for deglycosylating misfolded glycoproteins. And if this thing goes away, you might expect to see an accumulation of misfolded glycoproteins. And when you look in a liver biopsy report for Bertrand, that's exactly what you find. You find what appears to be an accumulation of these misfolded glycoproteins. And so at the time, the research team that gave us this data said, well, this is almost certainly, the answer to this, but there's nothing you can do about it. There's nothing actionable about this diagnosis, and there's still no way to prolong his life. That may have been true, may not have been true from their perspective, but I will point out right away that it did at least allow me to have another child safely. So my third child is here today, safe and happy and healthy because, somebody got the science right a long time ago. And so I remember writing the scientist the day he was born, the same ones who told me that it was not actionable, that actually it was, and that he's here because of that. I even wrote a paper about this whole process of family planning in light of a novel genetic diagnosis for other families that might want to do the same thing. But really what this did for me is it forced me in two different directions. One was into the basic science. And so because this is a glycobiological gene, I learned a lot of glycobiology and started partnering with researchers there to really understand the molecular mechanisms involved. I also knew I had to find other patients. So I wrote a blog post designed to do exactly that. You can see it there. This blog post was really designed to do two things. It had to go viral and it had to rank highly in Google search results. And so I made sure it did those two things. And this started a snowball in motion, led to an article in the New Yorker describing sort of the birth of this disease and the birth of this community as, in fact, patients did start showing up all around the world. And you can see me in the lower right there with one, every time I travel now, I'll end up meeting some of these patients anywhere in the world and stitching together this community. The community ended up enrolling itself in a newly created natural history study put on by the NIH. The New Yorker article led to my getting involved as first a consultant in the UDN, then eventually a co-investigator, this Undiagnosed Diseases Network. That turned into a faculty position at Harvard. And the underlying, basic science as it evolved turned into a treatment recommendation at about 2 years post-diagnosis. So 2 years after the diagnosis, I went through a little logic that said that, well, if what we have here is a missing enzyme, and an enzyme is something that turns A's into B's, when you take the enzyme away, you may either have too much A or too little B. And so there's a dietary strategy here. Can you give B? Can you restrict A in response? And there are examples of diseases where this works perfectly well. If you have PKU, you want to restrict phenylalanine, and if you have MPICUG, you want to give mannose. And it turns out NGLI1 fits this mold too. Through the basic science that was being conducted on the disorder, I got to have a sense of what exactly was accumulating and what exactly was becoming deficient. Turns out that what's deficient in NGLI1 is represented here by this little blue square. It is a sequestered metabolite. And this little blue square happens to be N-acetylglucosamine. And it didn't mean much to me at the time, but I was at least able to Google it and figure out that you could buy it on the internet. So I did that, ate an entire bag of it to make sure that it was safe to take. And after that, I woke up the next day not dead, and figured that was FDA phase 1 safety testing complete. Ended up ultimately, after a small amount of ethical wrangling, giving it to Bertrand, not knowing what it might do, but walking into his room a few days later to find that he was actually crying tears for the first time in his life. He was hungry waiting for breakfast, and he was actually making tears. And this was, the end of eye problems as he knew it for the rest of his life. He never faced another serious eye infection after finding this. And so, on a day-to-day basis, this is an enormous quality of life improvement for Bertrand. Went on to do a lot of other stuff after this. I ended up getting a grant to do novel drug development, working with planarian worms. We did genetic screens. We identified new drug targets. We did computational drug screens, and ended up identifying more therapeutics for this. We did virtual drug screens of 200,000 different molecules. 70 of them looked like inhibitors of this target we identified, and 14 were already FDA approved. And one actually worked when you checked it with mass spec, and that happened to be Prevacid. So Prevacid, became an important therapeutic part of his therapeutic regimen. And there's a lot of stuff in the middle. I'm running out of time, so I'll skip over some of the details. But yeah, I ended up going to the White House as a result of this to help engineer the precision medicine initiative for the entire country. Ended up at the White House for 3 years. It was a really interesting time for me. And, oddly enough, I got to transition. So at the end of my time with Obama, I could say, it looks like we've done initial clinical validation of the mission. I ended up sticking around for reasons I nowhere near enough time to explain now, for a year into the new administration. And, after a year is about all I could take. And so I quit. And people still ask me, what was it like, working for the Trump administration for a year? And all I can really say, I guess I can say a lot more now, is it was interesting on a day-to-day basis. And I gather remains so all the way to the very end. For Bertrand himself, he was happy for the vast majority of his life as a consequence of the many treatments that like the ones I talked about today, and several others that I haven't, that we're able to find for him. He became increasingly communicative, towards the last years of his life. He started using an eye gaze computer and was able to start speaking, really, conveying his sense of the world to us through that in a way that he never had before. So when I would come home from work, for example, he could say things like, hello, like Matt is my dad. Like he knew I was home. And this was his way of saying welcome. Unfortunately, he passed away recently in October, almost 13 years old, 10 years past his original life expectancy. So it's been, hard for me, as you can imagine, but also clarifying about my mission in life and the mission of the Institute. It's clear to me that this is what I will do for the rest of my life, is the work that he taught me how to do. And if you want to know more about, his life and his mission, I've put up a site, bertrand.mite.net, that talks about his life, but also one that I have transformed into an active catalyst for his legacy. So the algorithm for precision medicine lives at his site. It's a 90-page guide written for patients in the position that I was, essentially 13 years ago. And it talks about this step-by-step process that you can take. And it is the founding guide. It's, it's the evolution of the vision document that I gave to UAB when I came here 4 years ago, saying, this is how I want to structure this program. It's all up there for anybody to read at this point. There's also a little endowment for hope that's been set up in his name, and the interest on this endowment is used to help patients just like him. So I'll close with the very last patient, or the very first one to be helped by this particular endowment. It's an acanthosis patient that has this terrible scaly patching on their skin. Turns out through the undiagnosed diseases program here, they were diagnosed with a germline gain of function in EGFR, which we immediately realized we could treat with the existing EGFR inhibitors. We found a way to compound these, make them topical. Like, I'm brushing over a lot. It took us 2 years to figure out how to do this, by the way, but we did it, and it made a significant difference in the life of this particular patient. And the cost of the trial, medication for this trial for the patients has all been covered by Bertrand's endowment. So it's gratifying for me that his mission lives on in the form of the science and also in the form of direct aid to these patients just like him. If you're interested, again, please join us on a Monday. It's all by Zoom now, so anybody can drop it at any point and see how we do what we do.